Welcome back to Dr. Liu's VCE Biology. Today's topic is photosynthesis, cellular respiration, and biotechnological applications, as presented in the 2025 VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. Bioethanol is generated through anaerobic respiration, fermentation, within yeast cells. By utilizing biomass, such as waste starch, as a glucose source, the yeast operates in an oxygen-free environment to catabolize the sugar. This metabolic process must be maintained at an optimal temperature, approximately 25 degrees Celsius, which ensures peak enzyme activity and maximizes the rate of ethanol production. 1. Identifying the trigger. Phrase OK, the question says reduces the overlap of their leaves. This is a physical adaptation. If leaves don't overlap, it means the total surface area exposed to the environment is maximized. More surface area usually means more light capture. 2. Connecting adaptation to the rate of photosynthesis. Now, how does more surface area lead to a faster rate? I need to think about the light-dependent stage. More exposed surface area equals more photons hitting the chlorophyll pigments. If I have more light energy, I can split water faster via photolysis. 3. Narrowing down the biological site. I need to find the option that correctly links the input, light, to its specific location in the chloroplast. A and B talk about CO2 and glucose. Those are more related to the light-independent stage, stroma, not the primary thing captured by leaf surface area. C mentions oxygen, but oxygen is an output, not an input for photosynthesis. D says light on the grana. That's it. The grana, thylakoid stacks, are exactly where the chlorophyll is located and where the light-dependent reactions happen. Eliminate A. The consistency trap cells do not consistently yield 36/38 ATP. Different cell types, e.g., heart versus skeletal muscle, use different shuttle systems for electrons, and environmental factors change efficiency. Consistently is a red flag in biology. Efficiency varies. Eliminate C. The root misconception: root cells cannot photosynthesize, but they do perform cellular respiration. They receive glucose via the phloem from the leaves to power active transport of minerals. Eliminate D. The C3 vs C4 confusion. C3 and C4 are pathways for carbon fixation in photosynthesis. Once glucose is made, the way a C4 plant breaks it down for energy, respiration, is identical to a C3 plant. Identify B. This is a fundamental concept in the VCE study design. The shuttle cost of moving molecules across membranes means that theory is not equal to reality. Option B is the safest and most scientifically accurate statement. It acknowledges the tax cells pay for transport and membrane leakage. Vinegar production is a classic example of a cooperative biological process where the waste product of one organism becomes the fuel for another. Stage 1. Yeast cells perform anaerobic fermentation, breaking down glucose to produce ethanol and carbon dioxide. Stage 2. Bacteria. Acetic acid. Bacteria take that ethanol and oxidize it to produce acetic acid, which gives vinegar its sour taste. Correct answer, C. The Christie error. Option D. Bacteria are prokaryotes and do not have mitochondria. Therefore, they cannot process ethanol in the Christie. This is a common trap for students who forget that bacteria lack membrane-bound organelles. The plasmid error. Option A. While bacteria have plasmids, these are small loops of DNA used for horizontal gene transfer, like antibiotic resistance. They are not the site where glucose or ethanol is broken down. That happens in the cytosol. The lactic acid error. Option B. While some bacteria produce lactic acid, like in yogurt, the production of vinegar specifically requires an ethanol to acetic acid conversion. Option A, incorrect. Stage S is consistent with the light-dependent stage, as water is used to reduce NADP plus to NADPH. Stage T is incorrect. The light-independent stage, Calvin cycle, uses CO2 to synthesize glucose. It consumes ATP rather than producing it. Option C, incorrect. Stage W is incorrect because the light-independent stage does not produce oxygen. Oxygen is exclusively a byproduct of the light-dependent stage. Stage X is incorrect for the Krebs cycle. The conversion of pyruvate to lactic acid is the pathway for lactic acid fermentation, anaerobic respiration in animals. Option D, incorrect. Stage Y is incorrect because glycolysis is an exergonic process that produces a net yield of ATP, not ADP. Stage Z is incorrect for the Krebs cycle. While oxygen is the final electron acceptor in the electron transport chain, the Krebs cycle itself does not use oxygen as a direct input. Option B, correct. Stage U accurately represents the light-dependent stage. Within the grana, water undergoes photolysis, splitting, which releases oxygen as a byproduct. Stage V accurately represents glycolysis. This process occurs in the cytosol, where glucose is broken down into pyruvate, resulting in a net gain of 2 ATP molecules for the cell. The correct option is A. This stage occurs in animal cells. Logical justification. Stage X identification. The table shows the conversion of pyruvate to lactic acid. This is the pathway for anaerobic respiration specifically in animals and some bacteria. YB is incorrect. Lactic acid is not broken down into ethanol. In animals, it is eventually transported to the liver to be converted back into glucose, the Cora cycle. YC is incorrect. Unlike yeast fermentation, which produces ethanol and CO2, lactic acid fermentation in animal cells produces no carbon dioxide. YD is incorrect. All anaerobic pathways, including the conversion of pyruvate to lactic acid, occur in the cytosol, not the mitochondrial matrix. The correct option is B. Complete a literature review to identify the amount of NADPH produced in stage S. Option B, analysis. Stage S was identified as the light-dependent stage, water to NADPH. Since the rate of NADPH production is directly proportional to the rate of the light-dependent reactions, researching this value is a valid way to investigate photosynthetic rates. YA is incorrect. Rubisco is involved in the light-independent stage, Calvin cycle, but stage Z was the electron transport chain in cellular respiration. These are incompatible. YC is incorrect. Stage Y, glucose to ADP, describes the investment phase of glycolysis respiration. Measuring glucose usage here would investigate the rate of respiration, not photosynthesis. YD is incorrect. Stage W, CO2 to O2, is a biological impossibility. CO2 is consumed in the stroma to make glucose, while O2 is produced in the grana from water. A student should mentally sketch the graphs as they read the options. Light and CO2 both flatten out when the system reaches its maximum capacity. That's a plateau. Temperature is different. If it gets too hot, the plant breaks denaturation, so the graph goes back down. It doesn't plateau, it crashes. Therefore, 1 and 2 have the same up and flat trend, while 3 is the odd one out. The correct option is A, factors 1 and 2. Logical justification. To find the same trend, we must look at the shape of the graph for each factor over a large range. Factor 1, light intensity, and factor 2, carbon dioxide concentration trend, both show a saturation curve, a logarithmic style. Description: as the factor increases, the rate of photosynthesis increases rapidly at first. Eventually, the rate levels off, plateaus, because another factor, like enzyme availability, or the other substrate, becomes the limiting factor. Visual: an upward curve that becomes a horizontal line. Factor 3, temperature trend, shows an optimal curve, bell-shaped or asymmetrical. Description: as temperature rises, the rate increases due to higher kinetic energy. However, once it passes the optimal temperature, the rate drops sharply because the enzymes involved, like Rubisco, begin to denature. Visual: an upside-down U, or a peak followed by a steep decline. C3 plants, location. All stages of photosynthesis, including the Rubisco-led Calvin cycle, occur in the mesophyll cells. Timing: Rubisco is most active during the daytime, when ATP and NADPH are available from the light-dependent stage. C4 plants, spatial separation location. C4 plants use a two-room strategy to avoid photorespiration. CO2 is initially captured in the mesophyll, but Rubisco is located exclusively in the bundle sheath cells, where CO2 levels are kept high. Timing: Rubisco is active during the daytime. CAM plants, temporal separation location. Both initial CO2 capture and the Calvin cycle occur in the mesophyll cells. Timing: while they take in CO2 at night, Rubisco activity is greatest during the daytime. This is because the Calvin cycle requires the products of the light-dependent reactions, ATP and NADPH, to process the stored malate into glucose. One, the enzyme distraction affinity change. Think of the enzyme Rubisco like a worker who prefers to pick up carbon dioxide, CO2. However, Rubisco is easily distracted by oxygen, O2. The heat effect: as it gets hotter, the shape of Rubisco's active site changes slightly. The result: it becomes much more attracted to oxygen. This is called an increase in affinity for O2. Instead of making sugar, it starts wasting energy by grabbing oxygen. 2. The closed-door problem, stomata. Plants breathe through tiny pores called stomata. On a hot day, the plant is terrified of drying out, transpiration. The heat effect: the plant closes its stomata to save water. The result: CO2 cannot get in. O2, produced by the light-dependent stage, cannot get out. Inside the leaf, the CO2 levels drop while O2 levels spike. Rubisco is now surrounded by oxygen with no CO2 in sight, forcing it into photorespiration. Summary for students: the VCE buzzwords. In a 2-mark short answer, students should use these power words. Increased temperature leads to a change in Rubisco's affinity, favoring oxygen over CO2. Stomata close to prevent water loss, leading to a high oxygen or low carbon dioxide concentration inside the leaf. For students, Part CI is all about fair testing. In VCE biology, controlling a variable doesn't just mean keeping it the same, it means describing a specific action or piece of equipment used to ensure that only the independent variable, temperature, is changing. 1. Controlling sunlight, the energy source. The logic: if one plant gets more light than another, it will photosynthesize faster regardless of the temperature. We need to eliminate this noise. The method: use an artificial light source, like a LED grow lamp, set to a specific intensity. The precision: keep the lamp at a fixed distance, e.g., 30 centimeters, from each plant and keep it turned on for the same duration, e.g., 12 hours a day, using a timer. 2. Controlling water, the substrate, hydration. The logic: water is an input for the light-dependent stage. If a plant is thirsty, its stomata will close, which we know triggers photorespiration. We must ensure every plant has the same hydration. The method: use a measuring cylinder to provide a fixed volume of water, e.g., 50 milliliters, to each plant. The precision: water them at the same time of day and ensure they are in the same type of soil pot so the drainage is identical. To get the full marks, students should use the word fixed or constant followed by a unit of measurement. Bad answer: give them the same amount of light. 0 marks, too vague. Good answer: place all plants under the same light globe at a fixed distance of 50 centimeters for 10 hours a day. 1 mark, specific and measurable. Variable 1, sunlight. The good answer: place all plants under an artificial light bank at a fixed distance of 40 centimeters for a constant duration of 12 hours each day to ensure uniform light intensity and quality. Variable 2, water. The good answer: provide a fixed volume of 50 milliliters of distilled water to each plant daily at 9 a.m. using a graduated cylinder to ensure identical hydration levels across all test groups. In a VCE exam, simply saying it changes isn't enough. Students need to point out that glucose is unstable and temporary. Reason 1: rapid consumption and storage. The logic: plants don't just let glucose sit there. As soon as it's made, it's either burned for energy or packed away for later. The high-scoring answer: immediate consumption storage. Glucose is a dynamic molecule. It is rapidly respired for energy, converted into starch for storage, or turned into cellulose for cell walls. Therefore, current glucose levels don't show the total amount produced. Reason 2: destructive testing requirement. The logic: to accurately measure the glucose inside a plant's tissues, you usually have to harvest and grind up the leaves. The high-scoring answer: measuring internal glucose levels typically requires destructive sampling, meaning the same plant cannot be measured continuously over time. This prevents the student from collecting longitudinal data to determine a precise rate of change. If the exam asks for a better dependent variable, always go for something that leaves the plant or is easy to see. Alternative 1: the volume of oxygen, O2, gas produced, easy to collect in water. Alternative 2: the rate of carbon dioxide, CO2, uptake from the environment. The presentation walks through key VCE biology concepts using exam-style questions. It focuses on photosynthesis, cellular respiration, fermentation and biotechnology, data interpretation, and experimental design. Each question is unpacked with exam logic, common traps, and high-scoring explanations aligned with the VCAA study design. 1. Biotechnology and fermentation. Bioethanol production occurs via anaerobic respiration, fermentation, in yeast. Inputs: glucose from biomass, e.g. waste starch. Outputs: ethanol and carbon dioxide. Conditions: absence of oxygen and an optimal temperature, -25°C, for enzyme activity. Key idea: fermentation is catabolic, breaks down molecules and releases energy. Vinegar production. A two-organism process. Yeast, glucose ethanol, anaerobic fermentation. Bacteria, ethanol acetic acid, vinegar. Highlights cooperation between organisms and reinforces prokaryote versus eukaryote differences. 2. Photosynthesis structure, stages and rates. Leaf adaptations, reduced overlap, increased surface area, more light absorption, faster photosynthesis. Light is captured in the grana, thylakoids, during the light-dependent stage. Oxygen is a byproduct, not an input. Factors affecting rate of photosynthesis. Light intensity and co-concentration. Show saturation, plateau, curves. Temperature. Shows an optimum curve due to enzyme denaturation at high temperatures. Therefore, light intensity and co-concentration have the same trend. Cellular respiration. ATP yield from glucose is not consistent, not always 36 to 38 ATP. Actual yield differs from theoretical due to shuttle systems, transport costs, membrane leakage. Root cells do respire using glucose delivered via the phloem. C3 vs C4 differences apply to photosynthesis, not respiration. Anaerobic respiration. Animals, pyruvate lactic acid, no co-produced. Occurs in the cytosol, not mitochondria. Yeast fermentation differs, ethanol plus co. 4. Identifying pathways and stages. Light-dependent stage. Occurs in grana. Water oxygen. Produces ATP and NADPH. Glycolysis. Occurs in the cytosol. Produces a net gain of 2 ATP. Krebs cycle does not directly use oxygen. C3, C4 and CAM plants. All use Rubisco, but differ in location and timing. C3, mesophyll cells, daytime. C4, Rubisco in bundle sheath cells, spatial separation. CAM, co-absorbed at night, Rubisco most active during the day, temporal separation. Emphasizes strategies to reduce photorespiration. 6. Photorespiration. In C3 plants, photorespiration increases with temperature because Rubisco's affinity shifts toward oxygen. Stomata close, lowering Co and raising O inside the leaf. Leads to wasted energy and reduced glucose production. 7. Experimental design and skills. Controlling variables requires specific, measurable actions. Light, fixed distance, duration, and intensity. Water, fixed volume, same time, same method. Using glucose concentration as a dependent variable is poor because glucose is rapidly used or stored. Measuring it requires destructive sampling. Better dependent variables. Oxygen production. Co-uptake. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.